Bismillahirrahmanirrahim. Assalamu alaikum. This is Madiha Sagir from Department of Zoology, Bank Road Campus, UAE, Lahore. So, students, today we are going to study about taxonomic procedures. As you know, that taxonomy is to classify the things, especially organisms, into different classifications, into different categories. So, the first step in the taxonomic procedures is the definitely is the collection of the specimens. So, today we'll focus on the taxonomic collections. Okay, once the very first step in the taxonomic studies is the collection of materials or the collection of specimens. The specimen word you have heard a lot. It means sample, right? I think you have heard a lot about the specimen or sample of the blood, sample of the tissue. So, in case of taxonomy, specimens represents or refers to the organism or part of the organism that is representing its own species. So, the very first and very important step in taxonomic study is the collection of specimen. But in order to start the taxonomic study, once you have a taxonomist have planned to collect the specimen, so he should not collect the specimen from a single particular area. To collect the specimen, you should visit as much as localities as possible. Why? Because if you are collecting your specimen or your samples from the diverse localities, then it will give you good results. But if you are, suppose I am a taxonomist, right? I have planned to collect the sample of or collect the specimen of butterfly. If I visit a very small area and I collect my specimen, but so what happened? What happened that the specimens from the other area or other localities, the butterflies of the other area may belong to the same species to my specimen, but because of the different habitats, they show the different morphology. They show the variation. So it will lead or it will pose the problem in taxonomy studies, in taxonomic collections, and it will finally and ultimately lead to the improper assessment of the taxa or improper assessment of the classification of that particular specimen which I have collected from a very limited area. So, for the taxonomic collection, one taxonomist should visit or should collect his specimens from as much diverse localities as he can. Seasonal variation, variation in the developmental stages, host parasite relationship, etc., should all be taken into account while collecting the specimen for studies. This is very important, as we have studied in our previous lecture about the variations. So, while collecting the specimens, the variations or different variations should be kept in mind. Okay, students, as I told you that the very important step in the taxonomic studies is the collection. So there are different methods that taxonomist could adopt. First of all, when he is collecting the specimen from a particular area, what should he do? That he should collect the specimens of the related group as well. What happened, because related group specimens are also available when you are looking for your own specimen. Now what happened from this, you can give this related group specimen to an other specialist who is working on it. This will help you. You can give this specimen to the other taxonomist as a loan, or you can mutual exchange your specimens, right? He will give you your specimen if he has, and you are giving his specimen. So this will be, in this way both the taxonomists would be benefited. Secondly, once you are planning to look for the specimen or to have a specimen, to go for searching the specimens, a taxonomists should focus on those areas first that are more vulnerable to the destruction or to the damage. Because what happen, if the area will become completely damaged or destroyed, then all the organisms including plants and animals, that is fauna and flora of that particular area, would be destroyed, and they become extinct. So before their extinction or before their damage, it is very important that they should be collected by the taxonomist. So first priority of the taxonomist should be those areas that are going to be destroyed or going to be destructed, either because of the flood or because of other environmental factors. Where and how to collect. The choice of collecting stations should be planned carefully. Now, if you are working on the variations, especially geographic variations, the variations of the organisms that are due to their geographical distances or that are due to their geographical relationship or their environment, where two steps should be taken into mind. One is all the material already available in collection should be assembled and analyzed. Before working on the geographic variations, a taxonomist should collect all the already available material and analyze it. Now, what will happen? This will help you to study the further variations that are being present in that particular group or that particular population, that particular species. Now, the filling of the crucial gaps thus shown will be the goal of all subsequent collections. Now, what happened after the study of the already pre-assembled collections? What will happen? A taxonomist will get to know about the gaps that are present in the collection. So go for he will go for those species that fulfill these gaps. He will go for those collections. If species show noticeable geographic variation, the collection should cover the range of all the subspecies. Subspecies is the rank that comes under the rank of the species in the taxonomy. And definitely, if you are going for the geographic variations, then you should collect almost, your collection should be so wide that you can fill the gaps and you can cover the range of all the subspecies. If species show seasonal variation, now in case of seasonal variation, what happen? uh, you have to be focused on in the particular season for the collection of the specimen. Collection should be made in various seasons. In many invertebrates, the season during which sexual maturity occurs is very short. Okay, so in that season, at that short time period, collection should be made. Okay, collections should be made at that time when the specimens or organisms are present in their best condition. For example, if we talk about the birds, birds shed their plumage, they undergo through the process of molting, they shed their feathers as the season changes. Suppose from summer to winter, they shed their feathers and they got the new plumage of lighter color of white color. So collection of that particular specimen of birds should not be made at that time. It should be made after the birds have completed their molt. And if we talk about insects, then we can say that same for the insect. The collection of insects should be made at that time when they completed their ecdysis or their molting, means when they completed their shedding of the cuticle. If species wears or bleeds, collections should be made at the time when specimens are in their best condition. In birds, for instance, after they have completed their molt. Wears or bleeds means that when species are damaged, somewhat collection should not be made. Collection should always be made at the best condition of the specimen. As we talked about the insects, for example, has gone through many life cycles or many different stages of their life cycle. During metamorphosis, pupa stage, larval stage. So collections should be made for such kind of organisms that have the different stages of the life cycles. Collection should be made at all the stages of their life cycle. Okay, the next step in the taxonomic collection is the labeling. Labeling is a very important step, because without label, your specimen is of no use. Without accurate label, the specimen will be worthless. For labeling, what things should be involved or incorporated in the labeling of the specimen? First of all, the locality from where you have taken your sample or your specimen. You have to mention that locality or that area in your label. But besides locality, you have to add to all the information related to that locality. For example, you have to add the altitude of that locality, that how much its height from the sea level. You have to add the latitude, you have to add the longitude of that particular locality. Latitude is the angular measurements. Actually, these are the measurements of the area from the um, equator of the earth. Equator is the imaginary line that surrounds the center of the earth. So if we talk about the longitude, then it is the angular distance from of east to west, and if we talk about the latitude, then it is the angular distance of north and south with the equator. So we have to add all these information of the locality in our label of the specimen. Second, we have to add the status of the habitat, that what kind of the habitat, either it was a disturbed habitat or it was undisturbed normal habitat, or either type of the vegetation. If we talk about the animals, then we have to add the type of the vegetations or type of the plants that are present in that particular locality from where we are collecting our specimen, because most probably our specimens are fed on that vegetation that are present in the that particular locality. So type of vegetation must be the part of the labeling. Third is the type of the host. If we talk about the parasites, as you know, parasites resides in someone's, so we need to add the type of the host in case of parasites. If our specimen is parasite, then we have to add in our label the type of the host as well. So next, we are talking about the temporary labels. Now, what are the temporary labels? If I am collecting the specimen, if I am collecting my sample from the field, so for the time being, I have to make the temporary labels. Why temporary labels? Because when I am collecting too many specimens, maybe later in my lab, there is a chance of error that I will mix it up. So to avoid the error, we have to make the temporary labels. So temporary labels can be written in the field itself, and these can be replaced by permanent labels in the laboratory. However, care must be taken while changing the label, since it is likely to cause errors, right? The exact name of the locality is very important. This is what I told you previously as well. Okay, how the exact name of the locality should be done on the labeling. This is one of the example, or you can say this is one of the sample of the locality name in the labeling. There are many other ways also present to write the locality in the labeling in taxonomy, in the field of taxonomy. So let's take this example. If the locality is Muthanga, an area of Vianat Wildlife Sanctuary in Kerala. The collector is TC Narendan and date of collection is 2nd June 2019. So it can be written like this. Here you can see that the country name would be written in caps lock in capital India. Its area was which? Kerala. Vianad was the district, and locality name was the Muthanga, and collector TC Narendan. 2nd June. June is a 6 month, so it is written in Roman here, and the year is the 2019. All right. This what we can write the locality name exactly. But this is again, this is one of the methods. There are many other methods are also available in taxonomy to write the locality and to write the collector's name as well. Okay, if the locality from where you are taking your specimen is very small, or it is not well known locality, it is not found commercially, so what should be done? You have to add the name of the locality, but as well as you have to add the important landmark of that is present near that particular locality. You have to add that. So what you have to do, you have to add the name of the locality and its position relative to a well-known place should be added on the label. Like you are telling, if you are telling your home address to someone or your university address to someone, especially home address, so while telling the address, you always add a nearby landmark, right? that is nearby to your home. Same would happen in the case of the labeling, and that if the locality is not well known, what we do, we add the nearby known place into the label with the place with the name of the that locality. So, in addition to locality, other data is also needed on the label. For example, if we talk about the plants, then sometimes taxonomics add they record the sex of the specimen, and they measures the actual size of the gonads, the reproductive organs. They add these sizes on the label, as well as the degree of ossification of the skull. Why ossification of the skull is important? Because in this way we can determine the age of our specimen. Now, what is ossification? Ossification is actually the remodeling of the bones. Uh, Osteoblasts are being added on the bones to make them grow and to make them new. So, how much new cells have been added to the skull, or how much, what is the degree of ossification, from which we come to know that what is the age of our specimen. And weight is also important. Weight should be mentioned in grams mostly. Now, after collection and then labeling, the next step is the preservation and curation of the collections. Well, it is the duty of the taxonomist. It is his duty that he has to preserve his collection, he has to preserve his specimen, as well as he has to curate his specimen. Preserve, for further studies, and he should curate his specimen, that he is responsible for his specimens, that it may not be theft or it may not get lost uh, from where he has placed the specimen. So it is the duty of the taxonomist to preserve, and as well as to take care or look after his specimens or his collections. So for repositories and museums, this what I was talking about, a particular single taxonomist. When he is studying on something, that it is his duty to keep that something um, with great care. But if we talk about the repositories or museum, repositories mean the area where the massive data has been collected. So, in case of repositories and museums, special curators will be there to look after the collections. Curators are like custodians. Their duty is to clean the collections, is to look after the collection, is to protect the collection. This is in case of museum. This is the duty of the curators. This in museums, taxonomist is not responsible, but the museum curators are the responsible, or museum keepers are the responsibles. Like in many buildings, in many offices, we keep the custodians, we keep the curators that look after the building and building specimens. So in case of museums, same curators will be hired or are hired to take care of the collections. Now, preservation, is very important, because preservation of specimen is gaining more and more important, since they serve as a kind of ex situ conservation. When we think that some kind of species gonna extinct soon, so we need to collect the specimen of those species, and we need to conserve them or preserve them, because they will provide the ex situ conservation for that extinct species. Ex situ conservation means the same, if we talk about the wildlife, if we think that lion or tiger, elephant species are going to be extinct, so we take that to the zoo, for example. So zoo is what? Zoo is the ex situ conservation area. The species that is not present in its original geographic area, but we have preserved it somewhere else. So this what is called ex situ conservation, just to make them conserved so they couldn't be extinct, right? Since more and more species are becoming extinct, these preserved collections will serve as relics to study the species that one existed. Definitely, when species got extinct from the world, so what happened? That these conserved species, which we have conserved ex situ, will provide as a part or as a sample. Relics mean the same, as a part or as a sample of the those species that was once existed in this world. Okay, the methods of preservation curation differ from group to group. We never preserve insects in 100% alcohol. Alcohol volume should be very less because they make the insects dry, and the dried specimens wouldn't be of use. They will be worthless. Some insects are pinned and wings are spread if they are taxonomically important. Here I have given you the picture. These are the different sizes pins available for the preservation of the insects. And butterfly, moths, some species of grasshopper also pinned, like here. You can see the spread wings of the insect. So some invertebrates that are preserved, invertebrates could be preserved in that are preserved in formalin are very much perfect for study. So the method of the preservation differs from group to group. Okay, so far we have talked about the collections or taxonomic collections, then how and where to collect our specimens. Then we discussed about the labeling, um, including temporary labels. Then we talked about the preservation and curation of our collections. Now, next step is cataloging the collection. Now, now, cataloging is important. Why? When we have preserved the specimens, now we are going to place these specimens in particular museum or repository. So we need to catalog them. What is cataloging? Cataloging is make a list of items, right? So cataloging done in any case, in any store, in any repository. Besides the taxonomy, cataloging is a common practice when you have a large amount of items, so you need to catalog them, you need to arrange them into a cart, or you make a list of that items and you give them special numbers. So what is cataloging? Cataloging is a process by which all the parts of a specimen are linked to the information associated with them through a unique identifying number. Now, this identifying number is very important. This is known as the catalog number, by which number we can access the particular item or particular specimen. Depending on the group of organism, again, which kind of species we are talking about, which kind of group we are talking about, either they are insects, either they are parasites, either they are plants, either they are invertebrates, they are vertebrates. The cataloging is always differ for it differs specimen to specimen, or it differs group to group. For example, in higher vertebrates, an individual number will be given to each specimen, because in higher vertebrate the specimen is itself is a very large, so we give a particular individual number to that each specimen, and all the related information about that specimen will be given in the card, which will be filled in the catalog card, in card catalog. Okay, here I have used the different spellings of catalog, like here I have used you and here not. So this is not a problem. This is a difference between the American and the British English. You know that some American English you is not important, but in British we use you. Okay, in case of taxonomy, cataloging is usually done geographically. Cataloging usually done geographically, that is, all specimens collected at the given locality or district in a given period of time or by one expedition are entered in the catalog together. So now, what does it mean? That if I have gone to the traveling or to search the specimen, so whatever I got during that my expedition, I will catalog their specimens together in a single catalog card or single catalog number. So cataloging is usually done geographically. So specimens uh, are collected from a same area. Those specimens that have been collected from a given area, given locality at the same time would be cataloged together. All right, this is one of the way to catalog the things. Now this is done after authentically identifying specimens. Now cataloging is always done after the identification of the specimen. Once specimen has been identified, that it has been taxonomically classified to its genus and species level, so then it will be catalogued. Okay, otherwise it will remain under the study of the taxonomy. In groups where large numbers, in the previous slide I talked about that we can catalog the specimens on the basis of their groups. uh, Higher vertebrates are cataloged separately. Each of the catalog number or accession number would be given to each specimen, but in case of small specimens, it is not practicable to give a particular each number to every specimen. But what we do, we give the each number to a group of the collections. Like here I have written, in groups where large number of specimens are present, it is not very practicable to catalog each specimen, right? In the case of insects and similar groups, each number will be allotted to each set of collections. All right, each number will be allotted to each group of collections, each set of collection, not individual specimen, consisting of a set of specimens from the given locality or the area. The collections are usually arranged according to latest accepted classification. Science changes every day, right? So, what we do, we catalog the collection according to the latest acceptable classification, not the prior one. The latest classification of the taxonomy, the authentic classification, which is latest and acceptable classification, we arrange the specimens according to that. So unidentified, what happened? The unidentified specimens could be kept separately. They are not be the part of the catalog. They are not. They won't be part of our catalogs. Okay, here is the example of the vertebrates cataloging. What items or what important things that should be the part of the catalog. Number one is the museum accession number. In case of vertebrates, especially in higher vertebrates, we give each specimen to a particular number. So this is the that number that we need to access to our uh, specimen. This is the museum accession number or the catalog number. Second, original field number is also very important. It is also required. Scientific name of that particular specimen should be written while cataloging the specimen. Sex of the specimen, either it is a male, female, or intersex, whatever. So exact locality name should be mentioned from where it has been collected, and the date of its collection, collector name, that who collect this specimen particularly, and then the remarks on the collection or on the specimen should be the part of the catalog of that particular. Till now, we are generally talking about the collections. Now, the collections are further also divided into different types. We will discuss these types one by one. Number one is the survey collection. Survey collection depends on the surveys. It depends that we have taken into account a particular group that we want to survey on that, right? Again, I have taken the example of butterfly, right? If we want to study this group, we make a survey on this group. uh, or a survey could also be depend on the geographical area. So it depends that what group we have taken. Either we are working, or either we are making the survey on a particular group of animals or organisms, or we are working on the particular geographic area. We want to explore all the groups that are present in that particular area. So it depends, and those collection would be called the survey collections. Let's read this. This is usually based on surveys of a particular group or a geographical area. For studying biodiversity of a large group of a large area, it is usually necessary to devote more time, months, or even years for making satisfactory collection of the specimens. So here I have given one example. For instance, for studying the biodiversity of insects of Silent Valley National Park, if we want to discuss about the diversity of the insects that are present in that particular area, that is the Silent Valley National Park. If we want to survey the insects of the national park, then we what we do, it is necessary to collect almost all the orders of the insects which are present in area, and it will take years to identify each specimen up to the species level. So what we do in survey collection, first we decide that what we want to survey. Either we want to survey a particular area, or either we want to survey for the particular group of organisms. If we want to survey the particular area, then we will go to that area and we will look for all the biodiversity, suppose of insects present in that area, and it will take much time, because once we have collected all the biodiversity, then we have to identify all the biodiversity up to at least the species level. Now, generally, the biodiversity worker will be having a list of specialists. Now, if you are going on a survey for the collection of the biodiversity, you are not going alone, but you have a list of specialists that will work with you, so that your work will be prompt, will be quick, and will be more worthful. Generally, the biodiversity work will be having a list of specialists working in each group, because in bio, if you have selected the survey of a particular area, then you have, why do you need a specialist? Because you are a specialist in particular group, but in studying the biodiversity, you need to take all other related groups as well. So study the rest of the groups, you need the rest of the specialist of those particular groups. Since the success of any survey depends on accurate and fairly prompt identification. Definitely, your surveys for identification, so your success depend on the identification. This is the most important part of the work. Here is the second type of collections, that is collections for general exhibition. Now, collections that are meant for the general exhibition usually include the um, large or big specimens instead of the microscopic specimens. Microscopic specimens are never meant for the journal public exhibition. So, with the specimens that are being collected, or that are being displayed for public, we have labeled them with both scientific names and vernacular names. Vernacular names mean common names, and usually we used to display those specimens in the exhibition that are either very large, or that have some morphological description or difference with the other animals, so they would be more attractive to the visitors, right? Let's read it. Several museums exhibit various types of animals and plants for general public. Each item is identified, sometimes broadly classified. Okay, for general public, the items are not classified too keenly, but they are broadly classified. We have given that simple the kingdom name and the phylum name for the general public, and labeled with both scientific and vernacular names. Scientific names will also be there, and their common names will also be there. The names which we use in our common language. Vernacular names are those names. Usually, these exhibits may contain only morphological, peculiar, or rare or large specimens. In exhibition, we like to see something that is very different or very unique. So for exhibition, only those collections are displayed that have morphologically peculiar, means something different structures, right? Microscopic forms are not exhibited for general public. Okay, third type of the collection is a collection for the teaching students. These collection, most universities kept the collections for the teaching purpose for the graduate and postgraduate students. But these collections very limited. You cannot compare these collection to the repository collections, right? Usually they like to usually they like to keep only one or two items of a particular family to teach their students. So most university and colleges in underdeveloped or developed countries do not have trained curators. Well, if they do not, universities have don't have the trained curators or trained attendants for the specimens. So ultimately, what happened? Ultimately, all the specimens will be dried, and because of the lack of the proper care. Like in some universities, there are not proper attendants for the curation of the specimens. So what happen in these cases? These collections will be refilled by the students each year as a part of their curriculum work. because it is their assignment to collect the specimens and to preserve them. So in this way, these collections would be replenished. Okay, next kind of the collection is the identification collections. Now these are those collections that are met accidentally. Sometimes the officials are working or taxonomists are working on their own group of specimens. But what happen in that group? They may counter with a new organism, new unintroduced organism. So those specimens would be the identification specimens or identification collections. In quarantine stations, the officials then come across emigrant or accidentally introduced organism. Emigrant mean the species that is not inhabitant of that particular area, including most insects and some plants. So to find out the identity of this intercepted organism, what will happen? They call on for the a specialist, and then they do this task because they are working on their own groups. So they need other specialists to find the identity of that species that have been introduced accidentally. Some federal and state agencies found it necessary to set up centers for identification of insects because identification of insects is very intolerable. Because why? Because of their tremendous number. Insects are present in tremendous number or diverse number of species. There are too many insects. So to find out their identifications, government some in some areas government has made the setup for the identification of the insects, and there are two important necessary steps should be there. An adequate library should be present and a representative reference collection should be there. When there is a representative reference collection, then it would be easy to identify the identification collections, means the unintroduced introduced specimens. Okay, the fifth type of the collection is the research collections. Research collection is one of the most important collections because they provide the very important point or focal point for the taxonomic studies or taxonomic researches. So these kind of collections or specimens should be of very highest and finest type, and obviously for that they should be prepared and preserved with great care. These collections are very important, not only for the taxonomist that is associated with the particular museum, but it is also very important for the other institutions because these are those collections that mostly the two institution can mutual exchange, or they can take and give or this on a loan, in the way of the loan. So these collections are of great value not only to the museum taxonomists but also for the other taxonomists of other institutions, since the latter can get these collections on loan. Now, these collections where they must be kept. They could be under the custody of the taxonomist. It depends on the taxonomist, or they could be kept in private institutions, in public institutions, or in any university museum. Okay, sixth type of the collection or kind of the collection or specimens is the type collection. Type collection is the very important kind of the collection because collection is the physical evidence of particular organism. Type collection is a specimen of an organism to which a scientific name of that organism is attached. Or you could also say that type is collection, or type specimen is that specimen on which we defines the feature of particular taxon. So these collections are much more important. A new species is described based on a type specimen or the specimens. The type should be kept and curated with utmost care. Whenever a doubt arises regarding the identity of a species or genus, examination of the type is necessary to confirm its real identity. So these type of specimens are very important, and they should be preserved and curated very well. Why? For example, in during the Second World War, when German attacks Air Force attacks the Europe, the repositories, the type specimens that are present in the repositories of the Europe that have been transferred before the bombing to the underground cells, just to keep their types preserved and just to keep their types protected. All holotypes, lectotypes, neotypes, these all types should be preserved because they cannot be replaced. And the types should be numbered individually, and they should be kept as a separate collection in order to get special attention. And they should be facilitate for rapid transfer in case of emergency. This is what I told you in your previous slide. I gave the example of the Second World War. So types are customarily deposited in large collections, where they are deposited in large collections, and that repository or that institution is known as the standard type repository, where the large collections of the types are being deposited. In most repositories, if we talk about, they are catalogued alphabetically according to the genus and the species in case of invertebrates such as insects. And a card indexed by genera and other species are provided in order to save time in searching for the required type. This what you know already, that cataloging is important for the rapid accession to the particular specimen or particular kind of the specimen. Seventh type of the collection is the damaged collections or damaged specimens. So we could have the damaged collection because the damage has been occurred due to improper preservation processes, or if the specimens are not being looked after by the creators very well. So there is a debate that whether the repository or the museum should keep these damaged collections, or it's better to off these collections from the repositories or the museums. Well, the best way to remove the useless specimen will be to request a specialist who is working on that particular group will remove when he or she revises the group. Well, there is another point of view. Some people think, or some taxonomists think, that the damaged specimens should be kept in the museums. Why? Because whether it will prove useful one day when more and more techniques are developed to study the such specimens in future. So there is no harm in keeping the damaged collections as well in the museum or taxonomic repositories. So far we have discussed the different types of collections. Now it is the exchange of collections. Uh, this happens because collections are mostly exchanged between the countries. Sometimes the foreign country is not accessible to every taxonomist, so the different countries exchange their collections with each other. In order to build up a good collection of different groups, exchanges of specimens between museums and between specialists can be helpful. Sometimes what happens, the taxonomists have more than one specimen of the same group. So he may return, or he may give this specimen as a mark of gratitude or as a gift to the on the cooperation to the museums or other institutions. So exchange of collection is a good step in taxonomic procedures. It will help to build up a very good collection of different groups. Students, let's recapitulate our lecture, or let's summarize our today's lecture. It was about the taxonomic collections. So first of all, we talked about the methods of collection, then where and how to collect. From where we have to collect, we have to select the group, we have to choose the diverse geographical area or diverse localities. Then after the collection, we come to the labeling of the collection. First of all, we made temporary labels, then we replace the temporary labels by the permanent labels in our lab. Temporary labels are made in the field in order to avoid the error. Next, we go to the preservation and curation of the collection after being labeled. The After being labeled, the collections or specimens should be preserved with high care and should be looked after. Then in museums and repositories where large amount of specimens are there, we go for the cataloging the specimens and on the basis of basically on their geographical basis. And then kinds of collection, we come to the different kinds of collection, including survey collection, damage collections, type collections, collections for the teaching students. So next and last, we talked about the exchange of collection, that it is very good thing in taxonomy because it will help to make your collection at a very good level. To exchange could be done between the institutions, exchange could be done between the countries also. Okay, students, thank you so much and have a nice day, great day today.